In September 1928, Alexander Fleming returned from a holiday to his cluttered laboratory at St. Mary's Hospital in London. He had left a stack of culture plates on a bench. On one of them, growing among colonies of Staphylococcus bacteria, was a patch of blue-green mold, and around that patch the bacteria had dissolved. Fleming kept the plate. He grew the mold, identified it as a Penicillium species, and found that the liquid it produced killed a range of bacteria without obvious harm to animal tissue. He called the substance penicillin, published in 1929, and then largely set it aside. He could not purify enough of it to matter. The compound was unstable and present in tiny amounts. Extracting a usable quantity defeated several chemists through the 1930s. For roughly a decade penicillin remained a laboratory curiosity described in a journal almost nobody acted on, while people continued dying of infections that a spoonful of it would have cured. The question this leaves is uncomfortable. If a drug is that powerful, why did its usefulness begin eroding almost immediately? And why, nearly a century later, does the erosion look worse rather than better? The answer starts years before the drug ever reached its first patient. In 1938, at the Sir William Dunn School of Pathology in Oxford, Howard Florey and Ernst Chain went looking through the literature for antibacterial substances worth investigating. They found Fleming's paper. Chain, a biochemist who had fled Nazi Germany, took on the purification problem that had stopped everyone else. The Oxford team improvised. They grew mold in bedpans and biscuit tins, and built an extraction line out of milk churns, bookshelves and a doorbell. By 1940 they had enough purified material to test in mice. 8 mice were given a lethal dose of streptococci. The 4 that received penicillin survived. The first human trial came in 1941. Albert Alexander, a policeman with a severe infection, improved dramatically. The supply ran out. His doctors recovered penicillin from his urine and gave it back to him, and when even that was exhausted the infection returned and he died. Here is the detail that matters most, and it is almost always left out. In 1940, before penicillin had been given to a single patient, Edward Abraham and Ernst Chain published a paper describing a bacterial enzyme that destroyed penicillin. They called it penicillinase. Resistance was documented before the treatment existed. Nobody found this surprising for long. Penicillium mold makes penicillin because it competes with bacteria in soil, and it has been doing so for a very long time. Bacteria that shared that soil evolved countermeasures. The genes were already out in the world, circulating, long before any human isolated the drug. Production scaled anyway. American laboratories found a better mold strain on a cantaloupe in a Peoria market and worked out deep tank fermentation. By the invasion of Normandy in June 1944, the Allies had enough penicillin to treat the wounded on a scale that would have been unimaginable 4 years earlier. Fleming, Florey and Chain shared the Nobel Prize in 1945. In his Nobel lecture that December, Fleming spent part of his time warning about what was coming. He described how easily bacteria could be made resistant in the laboratory by exposing them to concentrations too low to kill them. He went further and sketched the scenario plainly. A man buys penicillin, takes too little of it, suppresses his infection without clearing it, and breeds resistant organisms that he then passes to someone else. Fleming said the thoughtless person playing with the drug was morally responsible for that death. The warning was accurate almost immediately. Hospitals began reporting Staphylococcus aureus that shrugged off penicillin within a few years of its widespread use. By the early 1950s, resistant strains were causing serious outbreaks in hospital wards, particularly in newborn nurseries and surgical units, where vulnerable patients were concentrated together. Chemists responded by modifying the molecule. Methicillin, introduced in 1960, was designed specifically to resist the penicillinase enzyme. It worked. It also bought less time than anyone hoped. In 1961, British researchers reported Staphylococcus aureus isolates that were already resistant to methicillin itself. That organism is now known worldwide as MRSA. It did not defeat methicillin by destroying it. It acquired a gene for an altered version of the protein the drug attaches to, so the drug simply had nothing to grip. The same principle underlies resistance to many later antibiotics. There are 4 main ways bacteria survive an antibiotic: and they are worth understanding because they explain why the problem is structural rather than accidental. The first is destruction. Bacteria make enzymes that chemically break the drug apart, as penicillinase does, before it reaches its target. The second is expulsion. Efflux pumps sit in the bacterial membrane and actively push drug molecules back out faster than they can accumulate inside. A single pump can often handle several unrelated antibiotic classes at once, which means one adaptation confers resistance to drugs the organism has never encountered. The third is modification of the target. Antibiotics work by binding something specific, a cell wall building enzyme, a ribosome, an enzyme that untangles DNA. Change the shape of that target slightly and the drug no longer fits, while the bacterium keeps enough function to survive. The fourth is reduced permeability. Gram-negative bacteria in particular carry an outer membrane studded with narrow channels. Shrink or lose those channels and large drug molecules cannot get in at all. Combined with efflux pumps, this is why infections such as Pseudomonas and Acinetobacter are so difficult to treat. None of that would spread quickly if bacteria only passed genes to their own offspring. They do not. Bacteria exchange DNA sideways between unrelated cells, a process called horizontal gene transfer, and it is the single most important reason resistance moves as fast as it does. The main vehicles are plasmids, small circular loops of DNA separate from the chromosome. A plasmid can carry a package of resistance genes and copy itself into a neighboring cell through a direct physical connection. A susceptible organism can become resistant to several drugs in a matter of minutes. Viruses that infect bacteria carry genes between hosts as well, and some bacteria take up loose DNA from their surroundings. The result is that a resistance gene which arose in a harmless soil organism can end up in a hospital pathogen on another continent within years. Selection pressure does the rest. Every dose of antibiotic that fails to kill an entire population leaves the survivors to multiply. Incomplete courses, unnecessary prescriptions for viral illnesses, and low doses given to livestock for growth promotion have all supplied exactly the conditions Fleming described in 1945. The scale of use is easy to underestimate. Global human antibiotic consumption has risen substantially since 2000, driven largely by middle-income countries where access improved. A large share of all antibiotics produced worldwide goes to farmed animals rather than to people, though several countries have now restricted growth promotion use. Measuring the harm took a long time, because deaths from resistant infections are recorded as deaths from pneumonia or sepsis. In 2022, the Global Research on Antimicrobial Resistance study published the first comprehensive estimate, covering 204 countries for the year 2019. Its finding was that about 1.27 million deaths that year were directly attributable to bacterial antimicrobial resistance, meaning the death would not have occurred had the infection been drug susceptible. Around 4.95 million deaths were associated with resistant infections in a broader sense. These are modeled estimates built from incomplete surveillance data, and the authors say so clearly. The uncertainty ranges are wide. What the study established is the order of magnitude, and by that measure resistant bacterial infection sits among the leading causes of death worldwide, comparable to major named diseases. 6 pathogens accounted for most of it. Escherichia coli, Staphylococcus aureus, Klebsiella pneumoniae, Streptococcus pneumoniae, Acinetobacter baumannii and Pseudomonas aeruginosa. The burden falls heaviest in sub-Saharan Africa and South Asia, where diagnostic laboratories are scarce and second-line drugs are often expensive, delayed or simply unavailable. The obvious answer is new antibiotics, and this is where the situation is weakest. Most major pharmaceutical companies left the field decades ago. The economics are hostile. A new antibiotic that works is held in reserve rather than sold widely, so the better it is, the less revenue it earns. The pipeline reflects that. The World Health Organization reviews antibacterial agents in clinical development each year and repeatedly concludes that the number in late-stage trials is insufficient, and that most are modifications of existing classes rather than genuinely new mechanisms. Truly novel classes reaching patients remain rare. Some progress exists. Public funding partnerships now support early development, stewardship programs have reduced unnecessary prescribing in several countries, rapid diagnostics can identify a pathogen in hours instead of days, and research into bacteriophages and antibody-based treatments continues, though none is yet a general replacement. The mold on Fleming's plate was already an old weapon when he found it, and the bacteria it killed had spent millions of years learning to survive things like it. What the past century changed was not the contest itself but its speed. We supplied the selection pressure, and evolution did the rest of the work.